Hello and welcome to Bio40 Pre-Lecture 13. Today we're going to discuss mutations. As with so many topics, let's begin with a whole lot of vocabulary. The term mutation itself has multiple meanings. It is both a heritable change in DNA and the process by which such changes occur. Genes undergo mutation, and then they have a mutation. When such mutation causes a change in an organism's phenotype, we call that organism a mutant. There are many ways to classify mutations. A mutation that changes a wild-type allele into something different is called a forward mutation. Remember that wild-type is not directly connected to dominance or recessivity. The new mutant allele could be dominant or recessive to the wild-type allele. There are also reverse mutations, also called reversions. This is when a mutant allele is changed back to the wild-type version. Mutations can also be classified as somatic or germline depending on what cell type they occur in. A somatic mutation is passed on every time that cell divides, but does not get passed to that individual's children. Only germline mutations are heritable across generations. We can also classify by phenotypic effects. You're already very familiar with dominant and recessive, but there are also terms for the magnitude of the effect. Null mutations, or knockouts, completely kill the gene function, while leaky mutants retain partial function. Some mutations are spontaneous, while others are induced by exposure to what we call mutagens. More on this later. It's important to note that these classes are not mutually exclusive. You can have a spontaneous forward, germline, recessive null mutation. Other types of mutations have to do with the molecular details of the change. Extra nucleotides can be added to or removed from the DNA sequence. These are called insertions and deletions respectively. In addition to direct problems caused by adding or removing genetic information, such changes can throw off how other bases in the gene are read, corrupting all the information that follows. This is called a frame shift. There are also substitutions, where one base is replaced with another. And there are a couple types of these. A transition is when a purine is replaced with another purine, or a pyrimidine with another pyrimidine. While transversion is a purine changing to a pyrimidine, or vice versa. Finally, we can classify mutations by their effects on the gene products. We'll discuss the details in the coming weeks, but most genes contain the information to make proteins, which are chains of amino acids. A base substitution in DNA can also cause a substitution in the amino acid chain, which is called a missense mutation. Additionally, base substitutions can cause the premature termination of the amino acid chain. That's called a nonsense mutation. While the missense mutation only affects a single amino acid, the nonsense mutation affects everything after it, so it frequently has a larger effect. In fact, many missense mutations have little to no effect at all. Some amino acids have very similar structures, and switching between them usually is not a big problem. This is called a neutral mutation. Finally, some base substitutions don't actually change the amino acid sequence at all, so the protein is completely unchanged. We call these silent mutations. Next, we have an example of the frame shift I mentioned earlier. Inserting or deleting a nucleotide alters how the gene is read, and alters all the amino acids after the mutation, which frequently leads to early termination as well. All of these mutations that result from single base changes to the DNA or single nucleotides being inserted or deleted are called point mutations. There are also large-scale chromosomal changes that can occur, and they're very important for evolution, but we'll talk about them later. I feel like there's a lot of previewing going on in this lecture. For a while, there was a debate over whether mutations occur randomly or are specifically induced. This is the difference between Darwinian and Lamarckian models of evolution. You may have heard which side won that argument. Darwin suggested mutations were random, but natural selection caused beneficial mutations to become more prevalent in a population. This is an important concept we will, that's right, discuss later in population genetics. Lamarck suggested a model where mutations occur in response to environmental conditions and are passed along to offspring. The classic example is giraffes stretching their necks to reach leaves and higher branches. In class, we'll discuss some important experiments which demonstrated this was not the case and that mutations are spontaneous. While Darwin didn't understand the molecular basis of mutations, he did understand that heritable changes are key to evolution. We now know that a mutation alters the DNA sequence, which alters a protein made by that gene, leading to a new phenotype. Beneficial phenotypes make the individual more likely to pass a mutated gene to future generations. But such advantageous mutations are rare. Most mutations are neutral. Our protein coding genes actually represent a tiny portion of our DNA. So most mutations don't even alter genes. Of those that do, many are silent or neutral and don't alter the function of the protein. Even some nonsense or frameshift mutations can have little to no effect if they occur near the very end of a gene. And most mutations that do have an effect are negative. It is typically much easier to break something than to create a new function. Because of this, cells have complicated mechanisms for keeping mutation rates low. There have been several attempts to get good estimates for the mutation rates in various organisms. One study examined forward mutation rates in mice using coat color. The researchers looked at 5 genes that affect coat color and saw 25 mutations in 2.2 million progeny. This is consistent with studies in other organisms that have found there to be 2 to 12 mutations per gene per million gametes. One important thing to note is that the mutation rates can vary greatly. Prokaryotes tend to have lower mutation rates than eukaryotes. But even within a species, you can see great variations in the mutation rates of different genes. We also see that forward mutations are much more common than reverse. You can break a gene by mutating it in various ways at various positions along its sequence. But reverting a broken gene requires specific mutations at specific locations. There are many reasons that different genes can have different mutation rates. Gene size can play a role. The larger a gene, the more bases there are to be mutated. Some DNA sequences are particularly susceptible to mutations. These are called hotspots. Different organisms also have different effectiveness in their DNA repair processes. The difference between eukaryotes and prokaryotes can be explained by the number of divisions. In a single-celled organism, one division is one generation. But in multi-celled organisms, there are many divisions that occur between when a zygote is first formed and when that individual is ready to make gametes of its own. And each division is a chance for mutations to occur. It's also been hypothesized that diploid organisms are more tolerant of mutations than haploid organisms because they have 2 copies of each gene. This all isn't to say that high mutation rates are always bad. Microbes growing under harsh conditions, and viruses like HIV that are constantly battling an ever-adapting immune system, tend to have higher mutation rates. The combination of large numbers and adverse environments can make mutations helpful. It's okay if most of the mutations are detrimental, as long as a small fraction get beneficial phenotypes that allow them to survive and reproduce. It's also a bad thing for the mutation rate to get too low. If there are no mutations, new traits can never develop, and organisms will be unable to adapt to environmental changes. There would be no evolution at all. Now let's discuss some of the ways that mutations can happen. Errors can occur during replication due to mispairing. We've established that A's normally pair with T's and G's with C's. But each base can exist in multiple forms called tautomers. Any given base can undergo tautomeric shifts, and when it's in its rarer form, it pairs with the wrong partner. So a large majority of the time, an A will pair with a T. But if it happens to be in the rare amino form when the polymerase comes through during replication, it will pair with a C. Likewise, G has a rare enol form that pairs with T's. T's enol form pairs with G, and C's amino form pairs with A. Note that we still always have purine and pyrimidine pairing, so these errors will lead to transitions and not transversions. Let's look at how these mispairs become stable mutations. Let's say there's initially a GC pair at the middle position of this DNA strand. When it's time for replication, the G is in the rare enol state and pairs with the T. Or maybe the DTTP is in the enol state when it pairs with the normal G. Either way, we have a GT base pair now. The other strand presumably has no such problem. By the time the daughter cells are ready for the next round of replication, the bases are most likely back to their common forms. So the G pairs with the C, and the T pairs with an A. After 2 rounds of replication, 3 cells are normal, and 1 has a mutation. But every time that one cell divides, the daughter cells will both have that mutation. Another type of error that can happen during replication is looping out, or slippage. Say you have the same base repeating like all of these A's here. As the polymerase moves, the strands could slip and a base could loop out. If a base on the template strand is looped out, the new strand will have 1 less base than it should, a deletion. But if the new strand slips, it will have an extra copy of the base, an insertion. Once again, these won't be stable until the next round of replication. Also, this is not limited to single base repeats. 2 and 3 nucleotide repeats can also display such issues with slippage. This is an example of slippage with a trinucleotide repeat. Here we have the sequence GCC repeated 8 times. As replication proceeds, the new strand slips, and 6 bases loop out. All the other bases are paired as they normally should be, so replication can continue. But after the next round of replication, there will be 10 repeats of GCC rather than 8. Expanding the number of repeats like this can have major consequences. Here's a list of conditions associated with trinucleotide expansion, including Huntington disease. In these conditions, genes that normally have just a few repeats expand to several hundred or even thousand repeats. Other types of spontaneous mutations aren't directly tied to replication. One such type is depurination. In this case, the bond between the sugar and purine is hydrolyzed, leaving behind an apurinic or AP site. This is a big problem for the polymerase that has to copy the strand. It's forced to insert a random base opposite that position, so this can lead to transitions and transversions. Another spontaneous cause of mutations is deamination. When the amine group is removed from cytosine, it becomes uracil, and uracil pairs with adenine, so this can lead to GC to AT transitions. Now, uracil is not supposed to be in DNA, and the cell has mechanisms to detect and repair it. But if it's not fixed soon enough, after the next round of replication, the A will pair with a T. This is a normal base pairing, so the cell won't be able to tell there's an error, and the mutation will be permanent. Certain cytosines are modified with methyl groups, making 5-methylcytosine. And deamination is an even bigger issue with these bases. Deaminated 5-methylcytosine is just thymine. And since unlike uracil, thymine is a normal base, the cell doesn't know there's a problem that needs to be fixed. So 5-methylcytosines are hotspots for mutations. Lastly, we have mutations caused by oxidation. Our cells have lots of free radicals, such as superoxide, hydrogen peroxide, and hydroxyl radicals that can cause oxidative damage to bases. The most common form of damage is a conversion of guanine to 8-oxoguanine, sometimes called 8-oxo-G or GO. 8-oxo-G pairs with adenine, so it leads to GC to TA transversions. This is a serious enough problem that there are 3 separate repair systems just to deal with this one modified base. That does it for spontaneous mutations. Next time we'll talk about mutagens, things that induce mutations, and we'll get into how the cell repairs mutations.